The topic of discussion for this module is the myelinated and non-myelinated nerve fibers. Nerve fibers are covered within the sheath of lipid as well as protein substance called as myelin. And this myelin is formed from the sheath of cells surrounding the nerve. And they are different from the connective tissue coverings of the nerve, that is epineurium, perineurium and endoneurium. So, the sheath of cells are called as Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. This kind of myelin sheath containing nerves are termed as myelinated nerve fibers, and those which are unsheathed are non-myelinated nerve fibers. So the myelinated nerve fibers are white in color and owing to the lipid containing sheath. And what is myelinogenesis? Myelinogenesis is a process of stepwise myelination around the nerve fibers of the nervous system. This myelination of the nerve fibers helps in faster conduction of the action potentials across the brain and hence quicker response. So the myelination in early life, it begins in the third trimester of the intrauterine life and continues up to 40 years of adult life. And the myelination of the peripheral motor roots is completed within the first month of the postnatal life, and that is sensory roots completed by 6 months of age. And next is the myelin sheath. So in the peripheral nervous system, the axons of the nerve fibers are myelinated with Schwann cells, which provide structure as well as metabolic support to the nerves. The axons in the autonomic nervous system and small sensory fibers, especially pain fibers, are unmyelinated. Myelination begins near the origin of the axons and ends just before its terminal branches. An axon lying near the Schwann cell invaginates into the cytoplasm of the Schwann cell, and axon gets suspended by the folds of the cell membrane of the Schwann cell, and this is called mesaxon. The Schwann cell now rotates around the axon so that the mesaxon gets wrapped repeatedly around the axon, which forms the spirals around it. And the lipids are deposited between the adjacent layers of the membrane. And these layers of the plasma membrane, along with the lipid, that is sphingomyelin, together forms myelin sheath, making myelinated nerve fiber whitish in color due to the presence of this lipid. This myelin sheath insulates the axon from the external environment, preventing ion influx. On electron microscopy, the cross section of the myelinated nerve fiber shows the major denser and the minor denser lines. So the major denser line represents the fused inner protein layer of the cytoplasm of the plasma membrane, where the minor denser line represents the external surface of the adjacent plasma membrane, which is made up of lipid. So the outside of the myelin sheath, a thin layer of the Schwann cell cytoplasm and the external lamina persists, forming the neurolemma. So each Schwann cell provides the myelin sheath for axon only for the shorter distance, following with another Schwann cell wraps the axon in a similar fashion. Hence, an axon in the peripheral nervous system is related to the larger number of Schwann cells along its length. So because of this myelinated nerve fiber is segmented at regular intervals with a short gap in between the Schwann cells, termed as nodes of Ranvier, and the part of the nerve fiber between these nodes is called as internode, and the length of the internode varies from 150 to 1500 micrometers. And what is the composition of the myelin sheath? The myelin contains proteins, lipids and water. The lipids include cholesterol, phospholipids and glycosphingolipids, Schmidt-Lantermann clefts. These are the oblique clefts which are seen in the myelin sheath, and these are thought to be formed due to the failure of the fusion of the adjoining layers of the Schwann cell's plasma membrane and leaving behind a layer of cytoplasm that passes spirally around the axon near the major dense line, forming a spiral space through which the perineural space communicates with the periaxonal space. And what is the clinical importance of this space? This space is an important passage for the substances from the external environment of the nerve fibers to the myelin sheath as well as axon. And the Schmidt-Lantermann clefts are greatly enlarged when the nerve undergoes Wallerian degeneration. And what are the functions of the myelin sheath? Myelin sheath provides mechanical support to the nerve fibers. It helps in increasing the conduction velocity across the nerve fibers when compared to that of the non-myelinated nerve fiber of the same diameter. It also provides insulation to the nerve fiber and also decreases the energy consumed during the process of impulse conduction. It is responsible for giving white color to the white matter of the brain as well as spinal cord because of the presence of lipids, and myelination in the central nervous system. The oligodendrocytes of the neuroglia are responsible for the myelination of the nerve fibers in the central nervous system. They follow similar process of myelination as that of the peripheral nerves, but a single oligodendrocyte provides myelin sheath around the multiple axons, unlike that of PNS. But if you see the non-myelinated nerve fiber, these nerve fibers are devoid of myelin sheath but are surrounded by Schwann cells differently from a myelinated nerve fiber. Here, the cytoplasm of a single Schwann cell is invaginated longitudinally by the several axons, and each fiber is embedded in a groove of the Schwann cell. So this is what you need to know about the myelinated as well as the non-myelinated or unmyelinated nerve fibers.